Hey everybody, today we're going to be learning about this process called PCR, or polymerase chain reaction. And this is used in one of two different scenarios. One is in crime scenes. So in crime scenes, a lot of times very little DNA is left behind. Maybe a tiny speck of blood, or maybe some other biological evidence like semen or hair follicles or skin cells. Either way, we're going to have to remove that DNA, and there's just not a lot to work with. So polymerase chain reaction is literally just making copies of DNA. Okay, so we can run this reaction and just create a ton more DNA to work with. And if you've ever seen crime shows, courtroom cases, there's a lot of convincing that has to happen in order to trust DNA evidence. So you don't just want to have just a few cells worth of DNA. You want to have as much possible in case something happens to your samples. All right, so we're just going to make more DNA from the crime scene. Another way we use it, so that's making copies of DNA, just simple copies, that's for crime scenes. Now, sometimes we actually want to check for the presence of a gene. Okay, so let me write this down. Okay, for instance, they found out there's a breast cancer gene. It's called BRCA1, and everybody has a certain form of it. Some people have a mutated form, but let's say if we wanted to check to see if chimpanzees, for example, one of our closest relatives, actually our closest relative, we wanted to see if they have a BRCA1 gene. So we could run PCR and check to see if they have it. And what's going to end up happening is we can target this sequence. So we can see if we took all the chromosomes, all the DNA from a chimpanzee, could we make a bunch of copies of this gene? And if they have the gene, the copies are going to be made. If they don't have the genes, no copies would be made. Okay, and I'll explain this a little bit more, but basically what happens is we put our DNA into a little tube in this machine. This machine's called a thermal cycler. Okay, and basically what's going to happen, along with the DNA, we are going to put a few different things with it. We're going to put a primer in there. So if you remember when we copy DNA, we do primers. We use primers to kind of help give the enzymes a place to land on the DNA. So we're going to add primers. We're going to add the sample DNA in there. We got to give it some nucleotides, because what we're doing is we're making copies of the DNA, so we have to provide it the building blocks. So nucleotides, so this is basically we're adding in A, T, G's, and C's. And there are literally biotech companies who definitely just specialize in providing scientists with just free nucleotides to use for this. The other thing we're going to add is some ligase, because we're going to have to basically put the primers, we're going to have to attach the primers to the DNA eventually. And we also need, normally our cells, when we copy DNA, we use DNA polymerase. This time we're going to use Taq polymerase, which is really just a heat-resistant form of the enzyme. And in your homework you'll see that basically this comes from bacteria that live in hot springs and really thermally active areas. And so we need an enzyme that's going to withstand the heat of this machine. And so that's why we get it from them. Our enzymes denature in that kind of heat. So this is just a special kind. So it's a heat-resistant form of DNA polymerase. Okay, so what this machine does, if we add in all that stuff, it's going to take our original strand. So remember, this is the DNA that's going to come from the crime scene or from the chimpanzee or whatever. And basically what it's going to do is it's going to copy that. And so the red DNA is the new strand. And basically it's just going to keep making copies. And you can see after 4 cycles, we already have 16 copies of DNA. And if this thing runs for 3 hours, we can get, hundreds of cycles in. And so you can imagine after, hundreds of cycles, how much DNA we can get. It's exponential. So here is what happens. And so I'll kind of draw this again in just a second, but what the machine does, it's very simple. It just cools down and heats up. So the first thing it's going to do, it's called denaturation, and it's going to heat up to 94. You don't have to know that, but 94 Celsius is pretty close to boiling. And what that does is that basically breaks the bonds between the A's, T's, G's, and C's. So it sort of artificially unzips the DNA, so we don't need helicase. Okay, so DNA comes apart. Okay, now the next step is called annealing, and this is the annealing of primers. And this happens at 54, so we cool the machine down, and it's going to basically allow the primers to stick. So remember, these are small pieces that bind complementary. And so if we're trying to check for a gene, we would make sure to add the primers that match with that gene. So this is the gene or DNA of interest. Okay, so if we're looking for the BRCA1 gene, we would just make sure we added primers that match that gene. And that way they would only stick if you had that gene. If you didn't have the gene, they would have nowhere to stick, and they would just kind of float around with nowhere to go, and no copies would be made. In the sense of crime scene DNA, this gene would be what we call STRs. STRs are short tandem repeats, okay? Short tandem, T-A-N-D-E-M, repeats. These are areas that are unique to each person, because most of our DNA is the same amongst humans, over 99% the same. So we really need to only copy the area that's going to be unique. Okay, and basically what's going to happen is the last part, it's going to heat up just a little bit more, and this allows our free nucleotides to bind. So it just happens to be a good temperature for our nucleotides to bind and our Taq polymerase to put on the new strands. Okay, so eventually all the copy is made. So let me just write that out again. Okay, so we're going to go about 95 degrees Celsius. That's going to pull us apart. Okay, then we're going to cool down. Then at that temperature, our primers are going to stick, right? Okay, and then lastly, at, what was that, 72, somewhere around there, what's going to happen is the rest of it is going to form. Okay, and eventually with some ligase, we're going to connect everything together and we'll have two copies. And so right after this, after the 72 degrees, it's going to heat, the machine will heat back up, and these will split again. It'll cool down, primers will attach, everything's going to go down over and over and over. So the thermal cycler just literally cycles between these three temperatures. Okay, and so once the extension portion is over, it'll go back to denaturation. And we can set it for however many cycles we want. Okay, so what does it look like when we're done? So let's say we were testing to see if chimpanzees have the BRCA gene. So here's what we do. We would take the DNA once it came out of the thermal cycler, and we would put it in a gel, okay? Chimp DNA. Okay, and we would want to make sure we ran a couple controls. So we would run, for instance, the human DNA here, because we already know humans have the BRCA gene. So we would see with the humans that we would get some amount of DNA in one of these slots. We're not sure how big the gene is, but let's just say it's roughly that big. If the chimp is positive for that gene, you should also see all those copies right here, okay? And so chimps actually do have that gene. So this is the chimps BRCA gene, and the humans would be there. And we would want to make sure we ran a sample of DNA that didn't have the BRCA gene, just to make sure everything's working okay. So let's say we ran a sea slug DNA. They don't have, obviously, a breast cancer gene. So we would see nothing there, okay? So no copies made here. So if we don't see any copies made, they don't carry the gene. They don't have that gene. Okay, pretty good. Now, in the case, and let me just erase what I have here. Oop, yeah, okay. Now, what would this look like in terms of a crime scene? Okay. Okay. So in terms of a crime scene, we would basically we would take the DNA from the crime scene, DNA from the suspects, okay, and we would run it through, and we would add restriction enzymes to the DNA that comes out. And so if the crime scene restriction digest comes out looking like that, suspect 1, they might look something like that, and suspect 2 comes out that way. All right, well, basically we know that it is suspect 2. All right, so it should match the crime scene's profile there. Okay, so this is called the DNA profile. Okay, so this is basically everybody's STRs cut with restriction enzyme. Okay, so we would make sure to cut all the DNA with the same enzymes and see how it turned out. And everybody's, like I said, should be different. Okay, so now you just have to do the worksheet related to this, and you guys got a quiz coming up on all those other genetics topics. So good luck on that. You guys got a lot of work to do. I'll see you next class to see what the results of our transformation were.